in one of the readings I set you at the end of last week, and you may not have had a chance to read it, and that's okay, but do yourself a favor because it's actually really interesting. David Esco and his colleagues talk about the idea that neuroscience and education do have these different goals. And we learned last week when we were listening to Imogen about how, you know, she was really reducing all these cognitive functions down to quite focused definitions and focused constructs, and then exploring, you know, how does this part of the brain be involved in this particular function. So that reductionist approach is really helpful when we're trying to understand how the brain is involved in particular learning processes. But as educational researchers, we're looking, or as educators, we're looking at the whole person. We're looking at their behaviour. We're looking at their interactions with other people. We're drawing on their prior knowledge and experiences. So we're embedding our learner in this socio-cultural context. We're not just reducing it down to these neurological processes. So our questions are different to what a neuroscientist would be asking when they're exploring different functions of learning. So we go about that process of understanding learning and measuring learning and collecting evidence of whatever the learning construct is in a different way to what neuroscience does. But there's sort of somewhere in between that we can also look at. And that paper by David Esco goes on to talk about how they have brought things like EEG out of the laboratory and into the classroom, so doing portable EEG. And Chase has been involved in that work within the Science of Learning Research Centre, but also works with other wearable devices so that we can capture physiological, outputs or insights into learning to give us that more of an insight into those subconscious or unconscious processes that are facilitating different aspects of learning. So that's what we're going to talk about today with Chase. So I'm going to hand over to Chase, and then we're going to come back and have a conversation a little bit more at the end. So Chase, I will stop sharing. And whilst Chase is setting up, I probably should introduce him first of all too. So Dr. Chase Sherwell, and Chase can also add in bits that I don't add in. So Chase and I have been working together for quite some time. We both finished our PhDs around the same time. Chase might have been a bit before me, and did his through Queensland Brain Institute. But Chase has done a lot of work with us in our school-based research, and is now we're working together looking at better understanding the processes involved in learning in MADA education, so in simulated learning environments. But I'm sure, Chase, you're going to talk a little bit about your research. Is that correct? Yep. So, yeah, that's right. So I will let you do that. Before I pass over to Chase, does anyone have any questions so far about kind of where we're heading with this conversation? Okay, all good. All right, Chase, I will mute myself.
All right, thank you very much, Steph, and thanks for inviting me along to talk today. I'm really looking forward to, discussing some of my own research, but also having a broader conversation generally about how, physiological research can help inform education research, but also vice versa. So just to give you an idea of where I'm coming from, my perspective. So I finished my degree in psychology, and afterwards I then went on to do, in cognitive psychology. So I'm very much on the reductionist approach, I suppose you would say. And after that, I did my PhD at the Queensland Brain Institute in cognitive neuroscience. So kind of just like Imogen. And that's me there looking younger and brighter with my EEG cap on. And so my research in my PhD was very much focused on looking at EEG and fMRI and those neuro methods, looking at, very small constructs, cognitive processes. And from there, I now work with Stephanie's lab, and I've worked with Stephanie for many years now, and looking at this kind of intersection of education and psychology to really understand the mental processes, the psychological states, and the behaviors that really lead to these desirable social, cognitive, developmental, and learning outcomes ultimately. But my real interest really lies in measurement. I'm quite obsessed with how we can measure these intangible processes and the best way we can go about it. So to give you an idea, this is like a very basic and simplified cognitive model of inquiry in psychology. And usually we start out with a context in which we believe a particular psychological phenomenon takes place. And the term context is very broad here. This could be an environmental context like the classroom. It could be a particular population like a clinical population, or the context could be we're looking at a particular intervention. We could be looking at a very particular cognitive task, or we could be looking at whether dispositional or trait factors play a role in different phenomenon, or in most cases, we're looking at some combination of these. And psychological research is really interested in the process that plays a role within this context that determines outcomes. When I talk about processes, again, very broadly, I'm talking about things like cognitions, affective states, decision-making, motivational states, you name it, things that are internal, kind of hidden processes that, as psychology as a field, we're interested in. And, these processes lead to outcomes. These are typically the things that we measure. So things like behavior, or you might, get participants or students to do an assessment. You might look at, objective performance on task. Things like reaction time is very common. Or in more social work, we definitely look at things like self-report measures, things like questionnaires and scales, but also like qualitative interviews are another form of outcome. And we're typically theorizing that, within this context, there is a process that underlies or determines the outcomes that we observe. And of course, in psychology, we test the role of these processes in many different ways. One way is experimentally by, perhaps putting participants into two separate groups, and then we expose them to different conditions, different contexts, or different tasks, perhaps different interventions. And then we, look at the different outcomes that occur in those two different groups. Or in terms of what we call observational research, we might measure several factors that we believe affect the central process of interest, and then measure outcomes depending on those different measures or the different factors that we measure at the beginning. Regardless, we're fundamentally interested in this underlying process, or what I sometimes call the mechanism of action that theoretically links these manipulations or predictive factors to the outcomes that we're interested in. And in almost all cases, we cannot directly measure that process of interest or that mechanism of action. Rather, we rely on quantifying these predictive factors or experimental manipulation in order to then infer the underlying processes based on the outcomes that we measure. And ultimately, this comes down to what we can readily observe or measure, as opposed to those things that we can't measure, that we have to rely on inference more to come up with our conclusions. And the inferences we draw obviously depend heavily on which measures we use, because we have to consider how these measures actually reflect the internal hidden processes of interest. So for example, here, this is just an example of a questionnaire asking about affective states. So, ratings on a scale of how distressed you are, how excited you are. And ultimately, there's a long chain of command that goes from the true underlying affective state all the way to the exact responses that someone gives on a scale. And this is just an example to illustrate that most of the measures that we use measure the end product of that entire chain of command. when it comes to things like self-report, there's a lot of things that can, interfere with ultimately the measure we have and the true underlying state. So there's things like self-report biases, whether or not participants can actually access or reflect on their own internal states. And this doesn't just apply to self-report measures, it applies to things like reaction time measures. There's a long, causal path that you have to go through to get all the way to a motor command or a reaction time. And this is no different as well for when we're doing observational research or even as teachers in a classroom. So, if you're observing a group of students who are working together and they're collaborating, if we're doing a scientific study on this, our observational measures, can be very subjective. We often rely on multiple experimenters to rate behavior, but also it's a subjective assessment of what we're seeing. And some work by our late colleague David Clark at the University of Melbourne, really showed that even when we're observing students in a classroom, expert observers, by the way, it can be really difficult to really gauge whether or not students are engaged in a task, whether they're participating, because although there's, very common and typical signs that we can look out for in terms of collaboration and engagement, it's still difficult to fully determine and rely on that kind of observation to get at whether or not students are engaged or actively collaborating. And this isn't unique to any particular measure. Every measure has advantages and disadvantages in terms of how accurately they reflect the underlying processes that we're really trying to get at. And this is kind of, a black box problem. We have to rely on inference, and ultimately we will have to rely on different strengths in the quality of our measures. But getting back to, tapping into internal states, one of the most common ways to do so is by using things like physiology and neurophysiology, brain imaging. So obviously in the laboratory, you would probably be familiar with things like EEG, also physiology in the lab, but also fMRI to really tap into the internal states of participants or students. But with advances in technology, there's also a lot of ambulatory devices. So ambulatory just means, you can walk around with them. So the most common ones are physiological devices. So things like wrist-worn devices that measure physiology like heart rate and electrodermal activity. But also there's now these really neat mobile EEG systems that can record brain activity from whole classrooms. And there's a number of advantages associated with these kind of more physical based measures. One of them, obviously, is that they're not very susceptible to response bias. So the things that we're recording are usually autonomous, automatic, so they're not under conscious control. So you typically won't see things like people, responding in a way they think you want them to respond. People generally don't have very good control over their underlying neural activity or their physiological states. Another big advantage is that you record a lot of data in real time. So it has a very high temporal resolution, and that is to say you get sub-second measures that are continuous over time, as opposed to surveys where you're really getting a snapshot. Here, you're more creating a film of people's states. And these states that we measure, changes in those states can reflect those internal processes and mechanisms that we're really interested in. And with these ambulatory devices, as the technology advances, they're relatively unobtrusive as well. I mean, one might argue that a EEG headset during a lesson is somewhat obtrusive, but students do get used to them very quickly. But especially with things like these wrist-worn devices, it's no different than a watch. They're not heavy. When we put them on students in our research, we essentially say, this is our version of a Fitbit, and they ignore them after about a minute. But one of the big advantages of being unobtrusive as well is that they don't interrupt the processes that we're interested in. If you are giving an interview or a survey to students asking about how they're feeling, how they're motivated, it causes them to reflect on those things. And in a sense, by measuring those things in those ways, you're interrupting the natural process. But using these devices, you are potentially tapping into these states without interrupting them as they would naturally occur. And that can be a huge advantage, especially in students. And that leads to one of my favorite topics, which is autonomic physiology. So when we talk about autonomic physiology, we're talking about activity of the autonomic nervous system, which is a division of the peripheral nervous system. So all the nervous system outside of the brain. And this system regulates our organ function by communicating between the brain and the spinal cord, which is our central nervous system. And the main role of this autonomic nervous system is homeostasis and allostasis. So homeostasis is making sure that all the systems in the body generally maintain a livable state. But just as important is allostasis. And this is adapting to the environment. So making sure that energy levels, etc., are in a state that will, keep you alive, but also ready to adapt and react to things in the environment, which is equally as important. Now, all of these systems are really connected to what's called the central autonomic network in the brain. And these are separate centers of the brain that are highly interconnected and generally play a role in things like goal-directed behavior, adaptability, emotion regulation, and homeostasis. So this includes a majority of the brainstem, but also emotional centers like the amygdala, hypothalamus, the prefrontal cortex, which is very critical for executive functioning, and also the hippocampus. So, why would we be looking at physiology when we're talking about mental states? And the main reason is because our perceptions, our emotions, and our cognitions both influence the state of the body, but are also influenced by the state of the body. So first, the mental processes affect our physical states, affect how our physiological systems function. And this occurs either directly through those connections coming from the brain to the different bodily systems, but it can also happen indirectly. So, things under our actual volitional control, like our respiration rate, how much we're moving around, these things are controlled by the brain and directly affect how our physiological systems run. But also in a very indirect route, enacting certain behaviors will have an effect on our physiological state. So in all these ways, this is mental processes in the brain that will be reflected in our physiology. But also the reverse is true. Our physiological states absolutely have an effect on our mental processes. So, how you feel about your current bodily state, this is something we call interoception. So, that sense you have of what your bodily state is can readily capture attention and can, shift the way that you're thinking. This often happens in a lot of psychopathologies, especially anxiety. Often people say that one of the first signs that they're feeling anxious is a bodily sensation. But also our physiological states can be integrated with our perceptions and cognitions. So what I mean by this is that, often memories will involve typical physiological states, but also your physiological states can affect how well you can perform certain cognitions like executive functioning. And perhaps also very critically is our physiological states provide a context for certain cognitive and emotional processes. And a major one is the encoding and recall of memory. So if you learn something in a particular physical state, your recall is actually better when you're in a similar physical state. And there's also central interactions between these kind of two directions. So often we will have representations of our sensory and motor states. So within our brains, we have representations of the external world, but also our current state. And we become very aware when changes in our state, changes in our bodily state are unpredicted and can really, shift our perceptions. And these reciprocal interactions between the mind and the body form the fundamental basis of psychophysiological research. We're really interested in identifying these bodily signatures of mental activity, again, as a type of outcome measure. So again, very rarely are we actually interested in measuring physiological processes as the process of interest. We're using these as a slightly different way of measuring the outcome to infer those mental processes that we're interested in. And when we're talking about the autonomic nervous system, it's really broken down into these two main streams that previously were very much viewed as antagonistic, although much more contemporary research shows that they can be highly selective and work in concert in many situations. However, largely they are viewed still as antagonistic. So the sympathetic nervous system, this is what primes our body for action and generally, increases the amount of available energy in the body to act. This is very frequently referred to as fight or flight responses, although I will have to say that is, really highlighting a very extreme example of the sympathetic nervous system in action. It is much more like a spectrum, and you can really think of it as the activating system of the body, which at any given time will be at some level. It's not just, panic mode. It's always on a spectrum. And this is really largely responsible for what we call increases in physiological arousal. So when we talk about physiological arousal, we are talking about that freeness of energy and the readiness for action. The kind of opposing nervous system is the parasympathetic system. And this is all about the opposite. It's conserving energy. It's what's often referred to as resting and digesting. So it is what's used for recovery. It's for digesting food. And when we see increases in parasympathetic activity, this is generally decreasing the physiological arousal. So it's slowing down that readiness for action. And when we talk about arousal, you will often hear somewhat interchangeably people talking about physiological arousal and psychological arousal. So physiological arousal is the physical state of action readiness, whereas psychological arousal refers to sensory and cognitive alertness. Now, in many cases, these two things are working in concert. So when you have, during wakefulness, for example, you have high physiological arousal and high psychological arousal. Likewise with attentional orienting, when something grabs your attention, you become more alert cognitively and in your senses, but also that will cause an increase in the action readiness in your body. Likewise with a threat response. So if you see something threatening, surprising, startling, you will become very alert and very ready to act. However, there are some notable exceptions. So with sedative drugs like alcohol, people can become very psychologically, alert and very active, while their physiological systems are kind of slowing down. We also see when people are experiencing delirium, we do see increased physical states of readiness. And also there's a very famous relationship between stress-related arousal and performance. So while people are very stressed, their body is typically very active in a very high state of physiological arousal, whereas things like cognitive performance and cognitive alertness can decline at very high levels of stress-related physiological arousal. So it's not always a simple one-to-one matching. And that's something that does need to be taken into account when we are trying to draw inferences based on physiological activity. So today I'm going to start with talking about sympathetic activity and how we can measure it. So one of the main ways that we measure this is actually by measuring skin conductance. So activity of the sympathetic nervous system results in the release of acetylcholine into our sweat glands, and this activates the pseudomotor nerves that fill our sweat glands. And as the sweat glands fill, they fill with electrolytes that increase the electrical conductivity of the skin. In other words, the electrical resistance is decreased. And this, by measuring this resistance between two electrodes placed over the skin, this gives us a measure of what's called electrodermal activity. And it really is just how conductive the skin is. And when we're talking about like sweating in this sense, you might think, oh, you get very nervous, you might get sweaty. But at any given stage, there is a very subtle level of how full your sweat glands are. It's not literally a difference between being bone dry and very sweaty. It is actually a very long continuum. And there's very subtle changes in the conductivity of the skin due to this sweat gland activity that does reflect generally the sympathetic level of the nervous system. If you have done any research into this area or read anything about this area, you'll first be kind of assaulted by all the different ways that this is referred to in the literature. So you might see things like electrodermal level, electrodermal response, skin conductance, skin resistance, skin potential, galvanic skin response. All of these things really refer to what generally now is called electrodermal activity. And the primary distinction here falls between what people refer to as like skin conductance levels and skin conductance responses. So if we look at the main signal here in the graph down the bottom, the combination of these is what we record. So if we look at this top line here, this would be the raw data that we record. It's just a signal of the current level of conductivity. But often what we do is we break this down into two main components. The first is the tonic component, and you can think of this as like the background level of sympathetic nervous system activity. It's very slow moving, and it's more reflective of like the homeostatic general level of the system. And it changes very slowly over time. And this is what we call skin conductance level. However, apart from this slow changing activity, there's these kind of rapid changing, spiking or peaking activity. And this is what we refer to as the phasic component. Phasic just meaning changing. And this can be further broken down into these individual peaks that we call skin conductance responses. And these are quick, fast, and reactive peaks that reflect reactions to the environment. So if you see a startling stimulus or have to think very carefully, these are rapid changes in your arousal level. Particularly, it's thought to reflect psychological arousal. So, if you're concentrating on a task or, you notice something in the environment that grabs your attention, this might be reflected in a skin conductance response. And experimentally, when we're investigating these skin conductance responses, we either refer to a defined stimulus that we know caused that response. So that's typically what you see as a skin conductance response. But otherwise, there's something called non-specific skin conductance responses. These aren't in response to nothing. This is just what it's called when we don't know what it's in response to. And these are actually the skin conductance responses that I'm really interested in my research. So in laboratory conditions, so experiments that are usually run in laboratories, you'll see people looking at individual skin conductance responses like you see here on the right. And the analysis of these responses will typically rely on extracting particular metrics, the most common probably being the amplitude or the size of that response. But that really relies heavily on, first of all, being able to measure a flat baseline where people are exposed to absolutely nothing. So just a baseline level. You'll also see huge differences between individuals. So you need to be able to record many skin conductance responses to similar stimuli, to understand what an individual's response looks like. But more importantly, you have to know what they're responding to. And you need to know the sub-second timing of stimuli. So that's why that type of research is typically restricted to the lab, where you have a very controlled environment where you can measure as many things as possible. And these kind of metrics are commonly associated with things like the intensity of emotional or affective responses to particular stimuli or situations, or even tasks. But importantly, it doesn't tell you about the valence of those emotional responses. So an extremely strong negative response will look the same as an extremely strong positive emotional response in terms of the sympathetic nervous system activity. Other things that it's commonly associated are things like fear conditioning. So fearful stimuli or threatening stimuli especially evoke larger responses, but also in difficult cognitive tasks. So things with a higher cognitive load will increase the size of these skin conductance responses as well. However, when we're taking this kind of these measures into real world contexts, so for me, that's typically classrooms, we have a lot less temporal precision. That is to say, it's a lot harder to know exactly when things are occurring to match it up with the physiological activity. And the major hurdle is we have zero control over what's happening in a classroom. And in many ways, we don't want any control. We want to know what actually happens in classrooms and how students, perceive and experience classrooms naturally. But that also means we can't really control what happens and can't manipulate things. And, a lot of our tight, nice control that experimental psychologists typically love goes out the window. But also, we're dealing with a lot of people at once, and there's going to be large differences between individuals. So because of that, we rely on kind of higher level or aggregate measures that really look at activity over time. Here, this is some data, excuse me, looking at EDA amplitudes of a class of 25 students. And on the left is all their data for a 60-minute lesson. This is just the raw data. And then on the right, we've zoomed in on the highlighted blue area when the students were doing a paired work task. And we've broken this down into those sharp responses at the top, that phasic component. And you can see they're all very short-lived peaking activity. And down the bottom is their kind of more background tonic activity. So that's the slower changes that you can almost think of reflecting their general alertness, their general arousal, whereas that phasic component is really their reactivity to the environment. And that could be reactivity to other students, to the teacher. It could be also reflective of kind of cognitive tasks like, okay, I need to focus. So it really is reflecting both external stimuli, but also what I refer to as internal stimuli or cognitions, affects, thoughts. So while a lot of researchers do look, take those tonic levels over time as their measures for investigating in real world contexts, what's becoming more common as well is actually looking at how often individuals have these skin conductance responses. And we can use that to categorize very broadly people's states as high, medium, or low arousal. So on the left here is that same image of the spiking activity. And then we transfer that into skin conductance responses per minute. So this is literally kind of like the speed or the frequency of their skin conductance responses. And then per minute, based on how many skin conductance responses they have per minute, we can categorize an individual as currently being in a high, medium, or low arousal state. Here on this picture on the right, they're coded with green, yellow, and red. And every horizontal line there is an individual student. And you can see, and to explain, the black parts are where the wristband has not made a good connection. So you do get some fallout in the data. But you can see there, like, very different patterns for different students. Some students have a lot of green, high arousal states a lot of the time. And then you see some students have a lot more red. You can also see that kind of yellow medium arousal is always only really occurring in short bursts. So it is considered more of a transition stage between the two. Excuse me. So when we're looking at these kind of measures, we're really looking at, trying to infer that link between the physiological and the psychological arousal. So here, when we see higher physiological arousal, we typically infer that this is related to greater amounts of attention and alertness, but you also see this kind of activity as in higher arousal or physiological activation, as it's sometimes called, when people are engaged in tasks, when they have higher cognitive load, but you also see it with higher task difficulty and higher stress levels as well. Typically, when you see low levels of physiological arousal or physiological deactivation, this is typically associated with disengagement from a task, boredom, essentially. But it can also represent when people are resting, conserving energy. It's not always black and white. It's not always a good thing to be in a higher arousal state, particularly not for a really long time. You do need those moments where you can conserve energy, reflect. You would never expect a student to be fully engaged, fully active for like an hour at a time. People do need to recover as well. And that's why it's interesting looking at how this changes over time. One of the key metrics that I've been using in some of my studies is looking at how long people spend in each of these arousal states during in learning environments and doing different learning activities. So one of the main measures I've been looking at is the incidence. And that is simply the percent of time spent in each arousal state, but also looking at things like how long these arousal states last for and how frequently people change between these arousal states. And about 4 years ago now, a study by Piera Diaz was tracked 18 lessons of a classroom, and they tracked 12 students and measured this incidence, persistent, and changes of levels. And one of their main takeaways from their study was that, surprisingly, on average, students spent most of the time in a low arousal state, and actually high arousal states, which are associated with engagement, were the least common. And this was, the authors said this was quite a surprising result because in their surveys of these students, they found that they were self-reporting as very engaged, very driven students. But one thing that we kind of observed was it was a very small sample size. And they also didn't kind of look at what students were doing in those lessons. So an honor student of mine, we looked at We tracked students over 6 lessons, but we looked at 318 students across 22 different classes. And we found a much greater balance between low arousal and high arousal states, which was really fascinating to find. But one of the key takeaways we found was that the more frequently that students were in high arousal states was correlated with their growth mindset. So we found that students that had that growth mindset were more frequently in high arousal states. And this is also particularly during group work. So we weren't particularly interested in when they were listening to the teacher. We were really focused on these work classes that were geared towards collaborative group work. And we found during that group work, the high arousal states were much more balanced with the low arousal states. So how
can I
do
ask a question?
Yeah, absolutely.
Can you just go back to that last slide? Because that's really fascinating, what you were just saying then. Can you just tell us, like you've shown us here, like your study, March 2021, but how did you test or measure their growth mindset? And how did you measure, like how were you capturing that they were doing group work at that time? Can you just explain to us what you were doing,
Yeah.
whether it's observations?
Oh, absolutely.
Yeah, thank you.
Yeah, so we recorded the classrooms for these lessons, and we specifically segmented the physiological data into the times when they were working with groups. So we extracted just that data based on our video footage of the students. And the growth mindset was from I think, it was a very standardized scale of growth mindset that they took at the beginning of the school year. I'll have to look up the name of that because it's quite a while ago that I helped work on this.
But that was it was a survey, like a self-report.
Yeah, just a self-report survey.
Yep. So you triangulated observations.
Yeah, observations, self-report, and our physiological data as well. We had a lot more data to work with. Like we had coding of like much more specific activities as well, but we just went very broadly, especially because, you know, with 22 classrooms, there's a lot of variability. So we wanted to purposely take a broader view rather than, you know, really breaking apart our sample into very small different tasks. But we felt that looking at, you know, 60 minutes of a lesson was too broad, and there's a big difference between listening to a teacher and doing group work in terms of the cognitive processes, the interpersonal processes, and of course the physiological processes that take part. And we've seen that in previous research, which I'll talk about shortly. But we thought, you know, group work at least, we're going to be looking at what we're interested in, which is, you know, when people are interacting and learning. Yeah, another question?
Yeah, may I ask, with that high arousal state there, you said that was during group work, and you concluded that there was more engagement. What about if, I mean, group work would also maybe lead to more social anxiety in somebody? So how could you, are there other measurements that you take to distinguish the two arousals?
Yeah. No, absolutely. And you're anticipating exactly what I'm about to talk about next, which is fantastic. It is like an alternative. However, what one kind of argument against that is that we then looked at how, like, when we say high, we're just talking about above a certain level. So we looked further at how high was, and they didn't really, when we're talking about high here, we're just saying over a certain level. Like anxiety levels are very, very high, and very rarely did the students actually reach that high. These are year 6 students as well. So these students knew each other quite well and were generally quite comfortable with each other.
Yeah.
But we didn't have any measures of affect in this particular study, which was a learning moment for us.
Thank you.
Oh, Oliver, did you have a question? Sorry, Mark.
Yeah, thanks, Chase. I was just wondering, with the sort of design, did you capture a baseline level of arousal for the individual students and then look at Okay.
Yeah, no, we didn't. These are very standardized levels, and it's a very good point you make about baselines. This method of looking at the peaks per minute doesn't while everyone might have a different overall arousal baseline in terms of especially the tonic kind of signal, this kind of formulation is kind of baseline free because it relies on the frequency of those responses, which shows a lot less variation between individuals, and that's why we chose it. Whereas if we were looking at, just these actual signals like the tonic activity, that baseline does shift massively between people, but the actual rate of response, rate of responding in those skin conductance responses is much less variable between individuals.
Awesome. Thank you.
Because we're looking at particular arousal episodes, and not just the overall level. That's a fantastic question.
Thank you.
So overall, when you're interpreting sympathetic activity, context is key, because sympathetic activity reflects a very broad range of processes, cognition, affect, a whole bunch, not to mention just physiological processes like sweating. So one of the main things to avoid is having a very narrow scope. And that's exactly what was brought up before, right? Oh, how do you know it's engagement and not anxiety or stress? And the only way to know that is by using other measures to provide more context to help your interpretations. Another thing to really look at, especially when you're reading research using sympathetic activity, is to understand the level of evidence. So, if you are reading a study that takes place in a lab and is looking at, individual skin conductance responses, you have to keep in mind that it is taking that reductionist approach. And while it might be very accurate of an individual process, it's unlikely to translate into, a classroom, for example, where you have 1,000 different processes and factors taking place. Whereas if you're looking at something that's more generally looking at arousal states, and perhaps the research is taking place in an ecologically valid environment, then you might be able to draw more realistic conclusions from that evidence. I've already touched on this, but again, I can't stress this enough. Sympathetic activity isn't just fight or flight. We need sympathetic activity to do anything. So, it's better to, rather thinking about sympathetic activity as, panic mode, it is a spectrum. It's about, free energy that we need to perform and live. But critically, we need multimodal methods or cross-validation to help interpret sympathetic activity. And like all physiological measures, these measures aren't here to replace other measures. They're not better than other measures like surveys or observation. It's just another tool in your investigative toolbox to look at things from a different perspective and to help, while physiology helps validate other measures are needed to help validate physiological inferences as well. I've noticed I'm going quite over time, Steph. Do you want me to just skip parasympathetic activity? I'll go over it quite briefly.
Just go over it briefly because it's really interesting.
So the opposite side, parasympathetic activity, it's innervated by the vagus nerve, the vagus nerve being the largest parasympathetic nerve in the body. I think it takes up like 75% of the parasympathetic system. But interestingly for us as psychophysiologists is that it innervates the heart. And at rest, the parasympathetic nervous system actually slows down the heart rate. But what's more interesting, perhaps, is that it increases how variable your heart rate is. And what I mean by that is it changes the heart from less of a tick-tock metronome and makes it more, the actual heartbeats themselves, more variable in their timing. So in this way, we can measure parasympathetic nervous system by how variable the heart rate is. The way we can measure this is by using electrocardiogram devices. And, you know, you might be more familiar with these in the hospital and such, but now we have these really neat little mobile devices. This is the one that I use. It weighs, I think, about 10 grams. Once you put it on, you essentially have forgotten that it's there. I wouldn't recommend using wristband devices for measuring heart rate, not for the accuracy that's needed to measure heart rate variability. You'll see, like, you know, your Fitbit isn't going to cut it for a scientific kind of inquiry into heart rate variability. There's a few different measures that people extract. The one thing you just need to know is that there's a whole bunch of different measures. They vary slightly, but not really enough to say that they're measuring different things. They all reflect this vagal activity, or the vagally mediated heart rate variability, which we are using primarily as a measure of parasympathetic activity. There's, again, tonic and phasic components to this, but they're slightly different. We're not breaking down a signal into two separate components, but they relate to how we measure it. So tonic HRV is your baseline heart rate variability. And that just means tonic here means you've recorded it at one time point. Whereas phasic heart rate variability refers to the change in heart rate. And when we're doing this in the lab, usually we use what's called the RRR paradigm. So it starts with resting, so doing nothing, relaxed, the baseline heart rate variability. Then we'll usually get people to be exposed to a certain stimulus or do a certain task, usually something that evokes stress or threat. And we look at the reactivity. So the change from the resting to the event is what we call the phasic reactivity. And then after the event, we get them to rest again, and we look at the change between the event and after the event. And this is the recovery. In real world context, though, you can do the exact same thing by just looking at heart rate variability before a critical event, during a critical event, and after. And currently I have a PhD student who is doing this exact thing in classrooms, recording students, particularly student and teacher interactions that naturally occur where there is emotion regulation involved. So usually these are students who have been identified as having difficulties in emotion regulation. And we're looking at both the teacher and the student heart rate before and after kind of teacher intervention that includes the student-teacher interaction. And this reactivity is really interesting because it reflects the kind of internal stress response to an event. And this has been looked at in many studies, and it typically is called vagal withdrawal. So this is when the vagus nerve pulls the brakes, essentially, off heart rate and lets heart rate increase, and it becomes more stable, so less variable. But what's really interesting is when we see this vagal withdrawal, usually you might think, oh, well, okay, decrease in parasympathetic activity means a large stress response, and that's typically viewed as a bad thing. However, we often see that the reactivity can be adaptive or maladaptive depending on the context. So if you're in a situation that requires executive functioning, so like an exam, a large parasympathetic, reduction, a large reactivity is a bad thing. However, if the stressor is something that doesn't involve executive functioning, it's actually advantageous to have a large response. It's kind of the difference between being stressed because you're going into an exam or being stressed because someone's broken into your house. And we often find that people who have better emotion regulation will show the adaptive response depending on the context. So it's not as simple as, big reaction is bad. It depends on the context. And finally, I wanted to talk about what's very close to me and Stephanie's heart, which is physiological synchrony. So this can be, physiological synchrony is the tendency of individuals who are interacting or engaged in a joint stimulus to show similar changes in physiology. And when we talk about physiology here, this can be electrodermal activity, it could be respiration, it can be EEG as well, or it can be heart rate. And there's many different ways that this is calculated, including like correlation, time-lagged correlation, and much more advanced kind of signal processing techniques. But it's a really interesting tool to look at how individuals might be sharing similar physiological, and the inference there is psychological states. And this could be between individual students, between student and teacher, or as a whole group as well. And not only can you look at this in snapshots, but this is some data that I recorded looking at groups who are sitting with each other in a classroom, and over time, how their synchrony changes. So in the top part, that's the student's layout, and we've just got lines showing the synchrony between individuals within their group. Red being very synchronous, blue meaning asynchronous, meaning their physiology is going in opposite directions. But the question people always ask me is, what does this actually represent? And one of the largest kind of traps or misconceptions is that this is some sort of biological process. And it's absolutely not the case that, if I have an increase in heart rate, it is somehow being transmitted to other people that I'm interacting with and increasing their heart rate as well. It's certainly not a process. This is really just a different type of measurement. I usually use the analogy that if you had two people and every week you got them to fill out, an anxiety and depression survey, if they were both increasing over time, you wouldn't say that those two people have some sort of connection or process going on. It's just the fact that they are sharing perhaps a similar process individually. So I usually explain synchrony as reflecting shared, temporally aligned states, which may reflect similar processes across individuals. But it does absolutely have a component that does involve kind of reciprocal interpersonal processes. So for example, if I'm talking to someone, there is a lot of body language, and these are all stimuli that people will show physiological responses to. And if you're interacting with someone, there are reciprocal stimuli. Just like another example is, if I start having a fight with someone and I'm yelling at them and they're yelling back at me, we're both experiencing a heightened affective state, usually a very intense affective state. So as you can imagine, our physiology would be aligning in the same direction. So in this way, it can represent interpersonal processes as well. But another misconception is that people say, was there synchrony or was there not synchrony? And it's not a binary present or absent thing. It requires a comparison. It is a kind of comparative measure where you say more synchrony or less synchrony. It's essentially how similar people's physiological state is. A very common thing that you'll see in the classroom is that when people are engaged in a similar activity, or attending to a common stimulus, like listening to the teacher, you'll see a large increase in synchrony. But with lab-based work, there's been a lot of work with teams, and there's a whole laundry list of things that are associated with increased synchrony. Things like greater comprehension between members in the team, but also when people don't like each other more, you see increases in synchrony. You see lower synchrony when people switch strategies. So if people are working on a task together and it's not working and they decide to adopt a new strategy, you see a large dip in the synchrony. And this probably reflects the fact that people are not aligned, they're not going through the same processes. You also see greater synchrony with greater team member interdependence. And there's a lot of studies that typically show that there's better team performance when you see higher synchrony. But of course, these lab-based studies often lack these prior interpersonal relationships, and those prior interpersonal relationships may play a crucial role in mediating these effects. And these effects are things that you absolutely do see in school-based synchrony studies, where you see that there's higher synchrony when students are engaged in collaborative monitoring. So during collaborative work, when they're not only self-monitoring, but monitoring the progress of other group members. You also see greater synchrony during difficulties in collaborative exam. But there's also very interesting work showing that there's greater synchrony between students and teachers, the more likable the teacher is as well. But a big problem with synchrony research is that there's a lot of conflicting results and a lot of null results in the literature. And I think a lot of this stems from the fact that there are so many different approaches to how synchrony is calculated, and there's a real lack of consensus on the interpretation of synchrony. And I think as a field, this is moving ahead, but I think currently it's a bit of a minefield trying to interpret all these different studies. So ultimately, I just wanted to end on, is physiology worth measuring, especially if you need all these other measures anyway to draw inferences? And of course, I'm going to say yes, but I think it's really most useful when you can use it to differentiate between ambiguous external states and processes. And it's also really useful for testing state changes without interrupting those states. And I think it's really useful for explaining internal mechanisms that lead to observable changes. And this really comes into a multimodal framework where there's complementary mutual verification of different factors that really provide this holistic view of learning spaces. And these multimodal approaches help resolve these ambiguities and issues that we have if we were just to use a single approach alone. But that's all I had to talk about today. Here's some references if you're super interested in learning more about physiology. But otherwise, I'm happy to answer any questions and open up to discussion.